And we are done for the day here. But at 7 o'clock at O'Malley on the Green, we have a wonderful party with a live band and special performances, celebration. That's starting at 7 o'clock. Our Cruising for the Cause. We always have a music event that goes with our Arts of Healing weekend. And this year it is that. So we're looking forward to celebrating with everybody tonight. Cruising for a Cause, O'Malley on the Green, 7 p.m. to midnight. There's going to be live music featuring the Yachtly crew and a performance by Golden Delicious. There's food and drinks. So get your nautical themed outfits on and come celebrate with us. Then tomorrow, Sunday, we will resume. In the morning, every year we have a memorial service for the women that we've lost, a sunrise memorial service. This will be taking place here at East Auditorium at 9 a.m. You're welcome to come if you'd like. It's also going to be virtual. And following that, we will have our closing session at 11 a.m. Where Dr. Carol Williams is going to be speaking about our Everest adventure, and then our keynote will be a longer talk, truly inspirational talk by Dr. Annie Almes on navigating life and death disparities healthcare disparities between Malawi and Alaska. So that's all. It's going to be in a different setting. It's going to be East Auditorium, which is just in a main hospital down the stairs. Please come. There's going to be a reception, and it's going to be really, really worth everybody's participation. So on to our closing session. We're saving, again, moving into some really important topics. Very grateful to the speakers today and to the speakers that are about to speak. Starting, we're going to bring back Dr. Megan Tenney, who's going to talk about antibody drug conjugates, which we've touched on earlier today as some of the newest form of therapies that we have to target the cancers that we're treating and to personalize the treatment that we're giving to the specific principles, biological characteristics of the cancers themselves. So this is the very essence of what we call targeted therapy. So thank you, Dr. Tenney, for speaking again. We look forward to what you're going to share with us on antibody drug conjugates.
Thank you. I think this is less of a daunting topic and I'm going to put this in my pocket so I can't drop it on the floor again. All right. So, thank you again. So I am going to be talking about antibody drug conjugates, specifically in GYN oncology. These again are my disclosures, and I will acknowledge Dr. Kathleen Moore, whose name has been mentioned a lot today as one of the smartest people probably any of us have ever met. And so she graciously gave me some of her slides because she is certainly at the forefront of developing these drugs and therapies. So a little bit of a background about antibody drug conjugates. What is an antibody drug conjugate? Well, as the name implies, it actually consists of three things. So first is a monoclonal antibody that is targeting some sort of specific antigen that is expressed on cancer cells and hopefully not on healthy tissue or on minimal healthy cells. Second, there is what's called the payload, which is hopefully a very highly potent anti-tumor agent that is specifically designed to induce cell death after being internalized and kind of discharged into the cell. And the third thing is what's called the linker. And this essentially connects the antibody to the drug payload. And the linker is very important because it needs to be stable because it's carrying around this super highly toxic drug and it needs to get it directly to where it needs to go. So it needs to be extremely stable in our circulation so the payload doesn't fall off and get where it's not supposed to be. And so this allows lower doses of drug to be directly delivered to where they need to go. And so how do they work? So there are a few steps. First, the antibody binds to the target antigen that's on the surface of our cancer cell. And that is the little Y-shaped thing. So that's our antibody carrying our payload. Then it gets internalized. The whole antibody drug conjugate gets internalized into the cell, and it gets shuttled to the lysosome, where it then gets released into the lysosome, and the payload gets out, typically. And then it enters the cytoplasm, where it then goes and affects its damage. Usually these are on the microtubules, or the DNA, and that results in cell death. And that is typically what happens with an antibody drug conjugate. So other things that I'll be talking about and other things you might read about or hear about when you're talking about ADCs, one is the DAR, that's the drug to antibody ratio. So that's how much payload is actually attached to each antibody. And it will be in a ratio of like 3-to-1. You'll see today up to 8-to-1. So that's 8 payload molecules attached to each antibody. So obviously the higher the DAR, the more drug that gets delivered directly into the cell. And then there's different linkers. There's non-cleavable and cleavable. Kind of what I described earlier is more of like the cleavable linker, where it goes into the cell and then the linker gets separated from everything. There are some non-cleavable linkers. I don't think any of the drugs I'm going to talk about today actually have non-cleavable linkers. And that doesn't get broken down quite as much. And so then the whole big bulky thing remains in the cytoplasm. And cleavable linkers allow us what's called bystander effect. So once the drug is separated and the payload is just floating around, in addition to affecting the cell and, poisoning the microtubules or something else, it can get outside the cell and then go kill neighboring cells that might not actually have the antigen on it. So they didn't get a direct delivery of the payload, but now that the drug is floating around, it can go and just kill neighboring cells that didn't necessarily have the antigen on the cell. So that's what cleavable linkers allow. So it can directly kill, and then it can kill surrounding cells as well. So it's a little bit more effective. So ADCs in ovarian cancer. We heard a little bit already, I think in multiple talks, mirvetuximab or MIRV was discussed. MIRV is an antibody to folate receptor alpha that has a cleavable linker, so it does allow for bystander killing. The payload is a maytansinoid called DM4, which is a microtubule disruptor. And the DAR is 3.5-to-1, so not very high. MIRV has been demonstrated in tons of studies to be safe and have a pretty favorable safety profile, mild GI toxicity, neuropathy, and ocular events, which I'll go into later. The first big phase 3 study called Forward 1, which randomized MIRV to chemotherapy, was negative. It showed no progression-free survival benefit. So everybody was worried that that was going to be the end of MIRV, but just like with what I talked about with the Milo study and the MEK inhibitors, doing secondary endpoint analysis and some other kind of post hoc things, they did see that in high folate receptor alpha expression, there was high favorability towards mirvetuximab. So this led to some different ways of how they scored things and the decision to move forward with kind of new scoring systems to better kind of detect who truly is high. And so Forward 1 was not the end of mirvetuximab. So the psoriasis study, this was the original study. This was the original next study in mirvetuximab, and it was for patients with platinum-resistant ovarian cancer, 1 to 3 prior lines of chemotherapy, and they had to be greater than 75% positive for folate receptor alpha. In 2+ in more than 75% of the cells. So it's now like kind of a dual staging. And most of the patients had 2 or 3 prior lines of therapy. So these were fairly heavily pretreated patients. And all of them had previous bevacizumab, and about half of them had previous PARP inhibitors. And so the results of Soraya, you can see here, the objective response rates were approximately 32%. And you can see that there were some patients who had a complete response, which in platinum-resistant ovarian cancer, we usually don't see. And then many patients had a PR or stable disease. And so the vast majority of patients did get some sort of clinical benefit. And the PFS was 4.3 months, overall survival 13.8 months. Over on the right, that's just pictures of what those numbers are if you're visual learners instead. So from a safety standpoint, there were no new safety signals that weren't seen in the prior studies. 86% of patients actually had some sort of adverse event, but most of them were grade 1 and grade 2, and only about 30% had greater than a grade 3 event. Importantly were the ocular events, which I'm actually going to get to a little bit later, which was not unexpected based on the original studies, and then some mild nausea, which also was not unexpected. So overall, it was pretty well tolerated. So the ocular events mostly were blurred vision and keratopathy, which is inflammation of the cornea. And it happens not immediately, usually after 1 or 2 cycles. Typically, it is low grade and reversible, and there was only 1 patient who actually had to come off of study because of the ocular toxicity. So it's very frequent, but it's mild and reversible and not permanent. And there were no permanent ocular sequelae. So after this study, we got accelerated approval, which we already talked about is kind of preliminary approval, waiting for a confirmatory study, which was just done. The MIRASOL study design looked at mirvetuximab versus investigator's choice chemotherapy in platinum-resistant ovarian cancer patients with high folate receptor alpha expression. I'm not going to go through all the specifics, but this was just published about a month and a half ago after being presented at the end of last year in Europe. And just like we saw with Soraya, there was a statistically significant improvement in progression-free survival with mirvetuximab compared to chemotherapy. Not huge, but it was statistically significant. And more importantly, like Dr. Hardesty talked about, an improvement in overall survival, which has never been seen before in a platinum-resistant ovarian cancer phase 3 trial. So this is now pretty much should be considered if the patient is folate receptor alpha positive. This is the adverse events. So as you can see, they're pretty consistent with Soraya. It's a small little chart. I know I'm not going to make you all read it, but there were very few adverse events that led to discontinuation of drug. That's the red arrow. And then the ocular events were similar to what we saw in the Soraya study. A lot of patients had it, but not many of them were grade 3, and none of them were permanent. And so this is just the packaging indication for Mirvetuximab, things to note is it does have the black box warning for the ocular toxicity, and I have a few slides at the end specific to ocular toxicity. And it is technically only FDA approved for platinum-resistant ovarian cancer as a single agent. And there it is in the NCCN guidelines. So other studies, Forward 2, which I think was briefly mentioned earlier as well, was done in platinum agnostic cancer, meaning it was platinum sensitive and platinum resistant cohorts. It showed activity across all folate alpha expression levels, including low levels, but higher response rates and higher expression. However, because of this study, because it did have response rates over all expression levels, the combination of mirvetuximab and bevacizumab, which is what this was, it was combination, is in the NCCN guidelines for both platinum sensitive and platinum resistant ovarian cancer with folate receptor alpha expression, with no quantification of how much the expression needs to be. So it's in the NCCN guidelines, but it's not an FDA approved combination. And then the current study ongoing is called Gloriosa. So this is a phase 3 study in the maintenance setting in platinum sensitive ovarian cancer. So looking at mirvetuximab plus bevacizumab versus bevacizumab alone as maintenance after patients have received platinum containing chemotherapy with bevacizumab for recurrent disease. So we look forward to those results. Next, on to cervical cancer. So the main ADC for cervical cancer is tisotumab vedotin, or TV, which is an antibody to tissue factor. It has a cleavable linker, and the payload is, I'm just going to say MMAE, which is a microtubule disrupting agent. This has a slightly higher DAR of 4.1 to 1. So the original TV study was GOG 3023. It was recurrent metastatic cervical cancer patients with 1 to 2 lines of prior chemotherapy after having progressed on platinum-containing chemo, plus bevacizumab if they were eligible. This looked at response rates for an outcome. About 60% of the patients were recurrent, so 40% were upfront kind of metastatic advanced stage. Others were recurrent, and most patients had actually only had 1 prior line of therapy. So for the results, again, on the right is just the graphic of the words on the left. So response rates was 24%, and 7% of patients actually had a complete response, which similar to the platinum-resistant ovarian cancer, for progressive, previously treated metastatic cervix cancer, complete responses are rare. And the disease control rate, which included our stable disease, was 72%. So the vast, majority of patients did benefit clinically from this. And the median duration of response was a few months. So progression-free survival was 4.2 months, which was significant. And about 30% of patients were still disease-free at 6 months, or still without progression at 6 months, which is fairly good. And then we do have overall survival data as well that showed overall survival of 12.1 months. And a 12-month survival rate of 51%. So half our patients were still alive at a year, which I also think is pretty good for our pretreated cervix cancer population. And safety-wise, similar to mirvetuximab, a lot of patients had adverse events, 93% of them. So everybody pretty much had some sort of toxicity. However, most were grade 1 to 2. And very few were greater than grade 3. specifically looking at ocular events. This time it's conjunctivitis, which is inflammation of the conjunctiva, which is the white of your eye. So different than the keratopathy or keratitis that's on the cornea. And then peripheral neuropathy and bleeding events. Those are the three things you kind of hear when you think about tisotumab. So the ocular stuff, and then specific to tisotumab is peripheral neuropathy and bleeding events. Again, these were usually low grade and did not lead to having to stop the drug, but just things to be aware of that are kind of unique to tisotumab. So after this study, just the same, it gets accelerated approval, and we move on to the confirmatory trial, which is GOG 3057 or Inova TV 301. Similar eligibility, tisotumab versus investigator's choice chemotherapy, which included some things that I can't remember the last time I gave, like vinorelbine or irinotecan. But this was the study design, randomized 1-to-1 to ADC versus chemotherapy. And the overall survival was statistically significant, 11.5 versus 9.5 months for tisotumab versus chemo. Additionally, progression-free survival was significant at 4.2 versus 2.9 months. Response rates were better in tisotumab as well. That's the top red box. And then the disease control rate was 76% for tisotumab versus 58% for chemotherapy. And all of those were statistically significant. And so this confirmed the initial accelerated approval. This is the prescribing information for tisotumab, and similar to MIRV, it has a black box warning for ocular toxicity, and then the additional warnings for the neuropathy and the bleeding events that I mentioned. And here it is listed in NCCN. So next, ADCs and endometrial cancer. We don't have any that are FDA approved. Now, there are NCCN listed ADCs for endometrial cancer, despite not being FDA approved. And there are some current studies that are going on. I kind of talked about HER2 earlier with our serous cancers. And so there are multiple HER2 targeted ADCs. The bottom TDM1, that's trastuzumab emtansine. That was kind of the first one that was tested in breast cancer and in endometrial cancers. I feel like it's kind of been replaced by the top one, TDXD, which has a DAR of 8-to-1 instead of 3.5-to-1. And so it seems to be more potent and just as tolerated. There's also some other targets to TROP2, which is, another molecule, or looking at folate receptor alpha in endometrial cancers to see if mirvetuximab or one of the multiple other newer folate receptor ADCs are effective. And then I just want to touch tiny bit about something that Dr. Hardesty, I think she might have even put up the same slide or something very, very similar, which I got from Katie Moore. So this is TDXD, or the trastuzumab deruxtecan. I specifically wanted to look at this trial, which is called Destiny Pan Tumor 02. It looked at multiple different tumor types, but specifically there were 3 GYN cohorts. Cervical, endometrial, and ovarian. And so this is the anti-HER2 ADC. And so initially when I made this presentation, I did not, the paper wasn't published, but it just was published. And so here are the final results for Destiny Pan Tumor 02. And the green ones are the GYN. And so you can see the tall bars are those who had a higher HER2 staining. The bar all the way to the left is all comers, and then it's 3+ and 2+. So the higher the HER2 staining, the more benefit, but there were good responses across all of our GYN tumor cohorts. And this is the progression-free and overall survival in those cohorts. A is endometrial, B is cervical, and C is ovarian. And so these are by HER2 staining. So pretty much across all of them, the top line is 3+, then 2+, then 1+. And so there did seem to be activity in all of our cohorts. And so because of this, TDXD is now in the NCCN guidelines for all 3 of our cancers that are HER2 2+ and 3+. It's hard to read. I know I had to fit all 3 on there, but it's there. And so that was just added this month from what I believe to the NCCN guidelines. So for all 3 of those GYN cohorts, so cervix, ovary, and endometrial. These are just some of the other ADCs that are under investigation that I'm not going to go into. But there's a whole lot of moving parts in the ADC world right now in our cancers and all other cancers. And then finally, I said I wanted to, if I have time, Joni, do I have like a few minutes? Just talk about the ocular toxicity, which is very unique to ADCs and requires very labor-intensive participation on the part of our patients. And so when we're talking about resource-poor settings and centering patients, this can be a challenge to patients who don't have access to ophthalmologists and the frequent eye exams that are needed. And so MERV, the ocular toxicities I said are in the cornea, which is, you'll see keratopathy in all of the studies. That's their generic word for all of the eye things. And that's because folate receptor, it's not on the ocular surface, but because of the linker and it's cleavable, so it gets off, and then they call it off-target delivery to the surface of the eye. So it doesn't have the folate receptor, but the bystander effect and the off-target delivery because of the cleavable linker leads to the eye toxicity. So it's just in the epithelium, and it is considered reversible, like I said, because the corneal epithelium regenerates. And so in the interest of time, I'm not going to go through all of these, but the blurred vision, keratopathy, dry eyes are the most common. No grade 3 or 4s, no retinal toxicity. And in the early studies, they kind of recognized all of this weird eye toxicity. And so they changed the way the MERV was dosed from total to adjusted body weight. And they came up with these mitigation measures, and that seemed to fix the problem. And so what do we have to do? So if you look on the left side, the eye care, so no contacts. They need an eye exam at baseline and before cycle 3, 5, and 7, and then as indicated. Patients have to use 2 different kinds of eye drops, both steroid eye drops and then lubricating eye drops that they have to start the day before and put on as many as 6 times a day of the steroid, but then they also have to do the lubricating ones 4 times a day. And so this is 10 times a day eye drops and all of these frequent eye exams. And so it takes a motivated patient to do that. But the good thing is, like I said, it's reversible, and you don't really have to stop for grade 1. You hold for grade 2 until it's down to a grade 1. But again, only 1 patient had to go off because of eye toxicity. And then for tisotumab, it's different. Here, the tissue factor is on the eye, and so that's why we see the toxicity, because it's direct toxicity. It's not the bystander effect. So it's highly expressed in the eye. And so that's why we see that in the conjunctiva. Again, it is pretty reversible. And once they realized the bottom graph, once they realized in the initial studies, they did their little mitigation and they found a significant decrease in all the ocular toxicities, including the conjunctival inflammation. And so this was pretty much on the other slide. So 53% of patients had the ocular toxicity, but again, they were typically low grade, reversible. And the longer you're on, it doesn't increase your risk. So it's not like 5 months and then, 10 months, your risk is twice as much as it was at 5 months. It's not dose dependent. So the risk exists no matter how many doses you get. And so this mitigation is a little bit different. They need an exam before every cycle. That is what the package insert says. I have a patient who has been on tisotumab longer than anyone else in the entire world, and she gets her eye exam. She's had like 40, 50 something eye exams. I can't remember what cycle she's on. She goes religiously. She does her eye drops because she doesn't want to stop, because she had 4 prior lines of therapy and has a complete response and no evidence of disease. She's a miracle, but this can work. So these people need 3 eye drops. So they need the steroids and they need the tears, but they also need vasoconstrictor eye drops that they have to put on while they're physically getting the drug, along with an eye thing that always has to remain cold. So they have to switch it out. Because the vasoconstriction is thought to hopefully decrease the amount of drug that actually gets to the eye. And so that's why that's different than the others, than the MERV. And this is just a little pictorial of what they need to do. And that's it. And yes, I got stuck in Fairbanks, but this is what I got to see when I was stuck